Welcome back, guys. We're going to cover Section 5 today. Sections 3 and 4 talked about the adaptive immune response and the parts of it. Section 5 is going to get into how it actually goes and attacks stuff. So that's what we're going to cover today. So we have our B cells and our T cells. There's an advantage and disadvantage of that system. The benefit of that system is with these millions of unique B and T cells, there's a high probability that an antibody or T cell receptor will bind to antigens and initiate an adaptive immune response. So there's a good chance you will have something to fight off a disease. The downside is, since these things are each unique, there's maybe only a few cells, okay, that will recognize any given invader. So you have a whole bunch of different ones, different types to check for, but there's only one or two of them. So if you get sick with something, it may take a while for these cells to reproduce at a level large enough to fight off that infection. So usually your body wins the race against the pathogens. When it doesn't is when you need to get medical help for things. But hopefully that point makes sense, that you got lots of different ones that check for antigens. The problem is that each one is specific, and when you encounter one that needs to get fought off, that one needs to reproduce a whole bunch. So when faced with invading microbes, there are two types of adaptive immune responses: humoral response and the cell-mediated response. The humoral response comes from, I believe, the Greeks and their understanding of the four fluids of the body, and I think that this one had to do with blood, if I remember correctly. Okay, so we're going to talk first about the humoral immunity and what that is. It's produced by antibodies dissolved in the blood. So we've talked a lot about antibodies. Now we'll get to see how they actually work. And I know there's not a picture on this page, but there's a diagram on the next slide that should clear this up for us. So in case you're not able to visualize what it's talking about. So humoral immunity comes from B cells, right? Since we're talking about antibodies, and B cells are the ones that make antibodies, and the antibodies that go into the bloodstream. So each B cell has its own unique type of antibody on its surface, okay? So here's the process. So step one, when a microbe enters the body, the antibodies on only a few of these B cells can bind to antigens on the invader. So only a few of these antibodies will bind. Antigen-antibody binding causes the B cells to multiply rapidly. So when it encounters something that when it encounters an antigen that actually binds, basically triggers those particular B cells to start reproducing a lot. Not other B cells, because remember, the other ones, they're unique to some other type of antigen. So it's no use in ramping up production of B cells that aren't going to bind to this antigen. So only the one that is actually bound is going to get reproduced, and it's going to get reproduced very quickly. So this is called clonal selection, because the antigen selects which B cells will multiply, and the resulting daughter cells are clones. So again, we're looking for the one that responded in particular, not the B cells that are making other antibodies. Those aren't going to matter. We're looking to reproduce the one that actually got triggered. Okay, so over here on the right side, we talked about these first three steps. So you've got these antigens out here, right? They're going to encounter the antibodies on these B cells, okay, which are in the dark blue. The antibodies are the things going around the outside of this. Notice we've got 1, 2, 3, 4, 5 different types here, but these antigens only bind to the one in the middle. Okay, so as soon as these antigens bind to this cell, this one is going to get triggered to reproduce a whole bunch because it's got the antibodies to fight this antigen. These other ones are still going to be floating around, but they're not going to be doing anything. They haven't been activated. Okay, so that was step one. Step two, the selected cell is going to multiply rapidly, so we're going to make a whole bunch of this one because it's recognizing that antigen. Okay, and then it's going to produce genetically identical B cells, which are going to have the same antibodies around the outside of it, and that brings us to step 4 here. The daughter cells differentiate into two types of cells: memory B cells and plasma cells. So if you've heard about plasma in blood, that's what it's talking about here. Memory B cells do not release antibodies but play a role in remembering the invader. So the memory cells, okay, are just going to stick around. They're not going to actually go fight stuff off. Okay. The plasma cells, though, they enlarge and fill with a lot of rough ER. So they're going to make a lot of rough ER, as you can see kind of in the picture here. Okay, these plasma cells make a whole bunch of endoplasmic reticulum. If you remember from last semester, that's going to basically be producing a whole, helping the process of producing a whole bunch of proteins. Rough ER, right? It's got a bunch of ribosomes on it, so it's going to make a lot of proteins. And then those are going to help produce antibodies, and those are going to get released out into the bloodstream. So these plasma cells, okay, are the ones actually making the antibodies. These memory B cells are going to stick around in case, in the future you get the same antigen, okay? So the secreted bodies have the same antigen binding site that was found in the antibodies located on the surface of the original parent B cell. So this cell, okay, these antibodies down here, kind of a hard little circle, okay, are the same ones that were on that original cell up there. Okay, so hopefully that makes sense as to how these things get produced. Okay, so there's 3 ways that humoral activity can shut down invaders. The first way to do this is actually for it binding directly to the molecule, virus, or cell and rendering it harmless. This is called neutralization. So this is when the actual antibody itself just stops the bacteria or virus from functioning as it's supposed to. All right, it doesn't even get a chance to do what it wanted to do. A good example of this is like snake venom. So snake venom has got a lot of enzymes in it, and if you've got the right antibodies for this, or anti-venom, right, which is what would be in there, some kind of antibody or something like this that would shut this off. Basically what happens is these antibodies bind right there and seal this thing off and stop it from working, okay? So that's how this one's going to work there, okay? Some viruses do the exact same kind of thing where they gain entry by binding to cell surface proteins, so on the outside of a cell, all right? If antibodies cover the viral proteins that enable this, the virus can never enter your cell. So if these antibodies, say this was a virus and they attach on right here, basically what that's doing is shutting off the virus from ever getting a chance to enter because it's blocking those proteins on the surface of the virus from interacting with the proteins on the surface of your cells. Okay, so step number 2, possibility number 2 for the humoral response. Antibodies may coat the surface of invading molecules, viruses, or cells, making it easier for macrophages and other phagocytes to destroy them. So the first one was neutralization that just shut it off. It stopped it from working. In this case, what's happening is, okay, you've got some pathogen. Okay, you've got a rough drawing over here. Pathogen that's got some sort of antigen sticking out of it here, right, is what that is. Okay, and if it triggers an antibody, all right, in our variable region, it's very particular, it binds just like that. What happens is actually the stem of this antibody acts as like a flag to flag down macrophages. It says, hey, come over here, this thing's bad. So you're like, well, what does the stem of a antibody actually do? Well, it's signaling for certain cells to come in and kill whatever this thing is or engulf this particular thing. All right, so it binds on here. That's the variable region. It's very, very specific. The stem then tells the macrophage, hey, come digest this thing. All right, and so then option number 3 for humoral response. When antibodies bind to antigens on the surface of a microbe, the antibodies interact with complement proteins that are always present in the blood. So these complement proteins punch holes in the plasma membrane of microbes, killing them. Other complement proteins make it easier for the phagocytes to ingest the invaders. So they just come along and punch holes in the microbes, and then it's easier to ingest them. Okay, so the humoral response has its limits. It's effective at fighting invaders that are outside of our cells, okay? And we'll see why here in a second. So antibodies are large. They're very large proteins. So they don't normally go through plasma membranes. Remember, the plasma membrane, the stuff that goes through it, it's small, it's uncharged, okay? These are huge. These are not going to just readily go through a membrane. So therefore, a humoral response is only effective against microbes or toxins when they are outside of a cell. So if they've gotten inside of a cell, all right, we can't have antibodies go get them, okay? Because antibodies can't go through there. So generally, this is in like blood, lymph, or interstitial fluid, okay, when they're outside of cells. So viruses are outside of cells. These are all susceptible. Antibodies floating around can attach on and identify these things as bad, and they can get, okay, digested. So bacteria and their toxins and some fungi and protists are usually vulnerable to humoral immune response because most of these aren't going to necessarily hijack your cells. They're just floating around in your bloodstream, okay? And so since they're exposed, they're not inside of your own cells, all right? They're pretty easy targets for this humoral immunity, okay? Viruses, on the other hand, require going inside of your cells to replicate. So again, talking about like why viruses are more difficult to deal with. This is a little bit of why they have to have, there's a different type of response that's needed. So viruses that are exposed to the humoral response when they are outside of the body's cells. So when this thing's replicating down here, and a whole bunch more of them are made, and this cell finally bursts, releasing a whole bunch of these guys into your bloodstream. For that moment of time, okay, those viruses, okay, are exposed, and they can get taken out by the humoral response, all right? But when they're actually still inside, these antibodies can't get there, right? So viruses are going to actually require the help from cell-mediated immune response. They're going to use the other response. Remember we said there were two types of responses here. There was humoral, and then there was cell-mediated. So a cell-mediated response is when the pathogen, usually in this case a virus, is inside of the cell, okay? Hopefully that makes sense. Things that are on the outside, humoral response can get to those. Things that are inside of cells, we're going to need this cell-mediated response. There's a particular type of cell called the cytotoxic T cells that deal with cell-mediated response. So cell-mediated immunity is produced by a type of T cell called the cytotoxic T cell, which attacks virus-infected body cells and cells that have become cancerous, which is actually kind of interesting. Your body does have a way to try to deal with cancer cells. It's difficult to stop them from reproducing, but they have ways to try to kill them. So anyways, here's the process. Cell gets infected by a virus. Boom. Some viral proteins are brought to the surface of the infected cell and displayed on the outside of the plasma membrane. So as this thing, okay, is getting replicated, some of the proteins right here, right, the identifying proteins might migrate and become part of the cell membrane. Hopefully you can understand that from the drawing there. Okay, so as this thing's replicating, some of the proteins wind up on the surface of this cell. Cytotoxic T cells in the bloodstream encounter the displayed viral antigens. They're coming along and they recognize, hey, that's not normally on one of my cells, so that's not right. These proteins form holes in the infected cell's plasma membrane, so the cytotoxic T cells come in and they're just going to remove, right, part of the cell membrane, okay? All right, and then they're going to release enzymes into the cell here, okay, to kill this thing off. All right, that's the idea. A very similar thing happens to cancer cells with cytotoxic T cells. These cancer cells will also get strange markers on the outside of their cell surface that aren't normally there, and your cytotoxic T cells recognize, hey, that's strange, let's go kill this thing. So similar idea happening to cancer cells. So what happens after a macrophage or dendritic cell engulfs a virus, bacteria, or some other type of microbe? It's got to get rid of it, and that's going to require the help of the appropriately named helper T cells. Okay, so you can kind of see the flow of things here. So we've talked about how these macrophages can identify the virus or bacteria or whatever, engulf it. Now it's inside, so what happens? Well, B cells and cytotoxic T cells cannot fight these invasions by themselves. They require the help of helper T cells. These cells have T cell receptors that bind to antigens displayed on the surface of dendritic cells or macrophages that have engulfed and digested invading microbes. Okay, so this guy, right, that's talking about, get a different color here. That's talking about, like, right there, okay? It's going to bind, this helper T cell is going to bind to surface markers on this macrophage or dendritic cell, okay? When its receptor binds to an antigen, a helper T cell multiplies rapidly, so this thing starts to divide a whole bunch, and that's going to enact this measure. Daughter cells differentiate and release cytokines that stimulate cell division and differentiation in both B cells and cytotoxic T cells. So basically, without cytokines, without this part happening, these cells' reproductive rates would be too slow to fight disease. Okay, so without this cytokine being released, we wouldn't get a whole bunch of B and T cells being produced. Now, this is part of a bigger picture, which you'll see here on the next slide. Okay, so this is a super helpful chart trying to keep all of this stuff organized. The first thing we talked about today was humoral immunity. This was B cells, okay, binding to an antigen, differentiating into plasma cells, which make a bunch of antibodies, and memory B cells, which stick around to remember what this particular pathogen or antigen was. We talked about cell-mediated immunity. This is when you have an infected cell and the virus is on the inside, usually viruses in this case for cell-mediated, and then the surface markers on the cell there migrate to the surface, the surface markers on this virus will migrate to the surface of the cell. Cytotoxic T cells recognize that, and then they're going to go off and develop into memory cytotoxic T cells, just like memory B cells, and then they also just kill off these infected cells, which you can see what's happening down there. And the last option was what happens after these macrophages or dendritic cells engulf the virus, bacteria, or some type of antigen. They will bind to helper T cells. Helper T cells then reproduce a whole bunch of B cells and cytotoxic T cells to go fight this. So without this step here, you wouldn't ramp up production high enough of those two things, right, that actually go off and fight stuff.